This is the Roots and All podcast, and I'm your host, Sarah Wilson. Join me as I talk about all aspects of gardening with some of the top horticulturists from around the world. Hello and welcome to Roots and All. This week's guest is ecologist and author Julian Doberski. If you think about compost heaps, how much do you really think about the living micro and macro organisms that dwell within them? How they contribute to the composting process, and how what you do to your heap can increase or decrease their chances of survival. Julian has written about the science of what goes on in a compost heap and about the living organisms who provide the hard graft of transforming waste into organic matter in his latest publication, The Science of Compost. I spoke to Julian to find out more.
Well, to begin at the beginning, I suppose, like for a lot of people, it was kind of related to lockdown. Because like a lot of people, I guess I was at a bit of a kind of loose end in terms of trying to find a project that would be useful and keep me interested. I had spent my working life primarily as a sort of ecologist, teaching ecology, which obviously involved stuff like soils. And indeed, we used to use compost to kind of demonstrate soil organisms and things like that. And so I thought, well, see if there was a market for a book on compost. Obviously, if you look at what's available in terms of compost, there is a lot, but they're a kind of different kind of book. I think most of the books which are on compost are written by people who have lots of practical experience of making weird and wonderful compost heaps and recipes for best quality compost and so on. So they're really practical guides on how to create a good, effective compost heap. What I thought I would do would be to try and fill in what I suppose I thought was a bit of a kind of gap in people's understanding actually what goes on in compost. Gardeners will put decaying plant material in at one end and expect to get compost out at the other end. But there's that kind of magic bit in the middle where I thought people might be interested to have some idea to what goes on. So that was really the driver of writing the book.
I suppose we ought to start at the beginning. So could you actually explain what compost is?
It's like a lot of terms, isn't it? They've acquired a kind of multitude of meanings. I mean, in the book, I do try to define what compost is, not in relation to, buying, say, a bag of compost in the garden centre, which, often in the past has been kind of peat-based plant growth material. I've obviously tried to focus on what a gardener would think of as compost in relation to running a compost heap, so that, you put your dead plant material in there and you get out something which is still organically rich and hopefully nutrient rich material that you have made yourself. So I guess that was the kind of definition I was using.
It is an interesting one. As you say, it's got a multitude of meanings. But one of the things that I picked up there when you were speaking is that you were saying about what goes in and then what comes out. And one of the questions that I always have about compost is what we put in effectively, I know obviously it undergoes a big transformation process, but effectively, does what go in then also come out? So thinking in terms of plant nutrients, for example, if we want a high nitrogen compost product at the end, should we use raw materials that are also high in nitrogen?
I mean, in general, of course, you would expect some sort of general relationship between the two. I mean, just as a bit of an aside, one of the things about compost is that, on the science side of it, you're talking about garden compost and garden compost making. There's actually very little published information on it. And you think, well, why is that? There are lots of people making compost in their gardens. I don't think anybody's got a precise figure, but RHS magazine came this week, which argued that there was something like 30% of gardeners make their own compost. And I have seen a similar figure in another publication as well. So lots of people are making compost, but everybody's compost heap is different. The mix of materials is different. In a sense, the nutrients that you start off with is different. What can you say about things like nitrogen? Well, I think it would be fair to say that there will be a general relationship, not that I've ever seen anything to indicate that in published form, between what goes in and what comes out. The key thing, or one of the key messages, of course, is that if you go to either side of the spectrum, if you have too much nitrogen, it's not good. If you have too little nitrogen, it's not good. So, somewhere in the middle is ideal. And of course, to get compost to start composting effectively, one of the things you would do would be to try and think about the material you put in relation to its likely nitrogen content, and where possible, increase that amount of nitrogen just to speed up, to make more effective the composting process, because nitrogen is very often, it's not the only limiting factor, but it's one of the main limiting factors on the speed of decomposition, because it's so key to life in general. No nitrogen, no protein, no cells, etc., etc.
Yeah, it's almost as if in gardening terms, there's quite often a deficit of nitrogen. It's the thing that we're all clamouring to get.
It's ironic, of course, because nitrogen is all around us. The air has got plenty of nitrogen in it, and yet we often talk about a deficiency of nitrogen. It's also ironic because in some ways, nitrogen is not only present as a gaseous form around us, but ecosystems, if we think in a broader sense, ecosystems are being swamped with nitrogen pollution. And some of them are suffering quite badly because of all our industrial processes, cars, et cetera, all pumping out various oxides of nitrogen and so on. Many natural habitats are actually getting too much nitrogen. But it is true that, that if you put dead plant material into your compost heap, which intrinsically has a low level of nitrogen, I mean, I suppose if you do something like cardboard or straw or whatever it is, that material is going to be very low on nitrogen. And if your compost heap is mainly made up of that kind of low nitrogen material, then certainly an additional source of nitrogen, assuming nothing else is limiting, that is a bit of an assumption. If nothing else is limiting and you add extra nitrogen, then you should speed things up. But I think the other point I made was that you can end up with too much in a sense. And, particularly if you've got a badly constructed heap of material in your compost, where you've got, for example, a lack of oxygen getting into the center of the heap. At the same time, you have a lot of nitrogen. What you may end up with is a rather stinky compost heap, generating ammonia, which will just disappear into the air. So, the issue of nitrogen is actually quite a complex one. And in the book, I try not to go over the top, because the whole process of nitrogen cycling is an involved and complex process. But I hope I give enough information to give a flavor of what is going on in what is really, as I said, a very complex process.
I always think in terms of compost, there obviously is a correct ratio of, for ease, we'll call it woody versus green material. And if you get that mixture wrong, as you say, it either takes forever and a day for it to rot down, or you end up with, like you say, a smelly grass heap. But either way, eventually, those things do seem to break down and reach an equilibrium. Is that the case? Is it just that it affects the amount of time that it takes for it to become compost?
Well, up to a point. I mean, if you had a situation where there was an absolute shortage of nitrogen, it will be a very slow process, because in a sense, what you're relying is on microorganisms which need that nitrogen to reproduce, to get involved in the actual decomposition process. In a sense, you can imagine, it's a very hypothetical situation, but you can imagine a lot of microorganisms doing some decomposition, in a sense, going through their life cycle, then dying off. And when they die off, that nitrogen is then available, I suppose, to the next lot of microorganisms, which live off their, the corpses of those microorganisms. So it becomes a rate limiting factor, the extent to which nitrogen, which is trapped, if you like, in the cells of these microorganisms, the extent to which that can be re-released and utilized by the next group of microorganisms, which will then carry on the decomposition process. But obviously, because you need that internal recycling of nitrogen to keep things going, that will in itself will slow things down.
Yeah. So apart from the microorganisms, are there other things at work in the compost heap helping to break it down?
Yeah, there are. I mean, it's a wonderful kind of model ecosystem. If we switch to think a little bit about an educational value of compost, it is actually quite an educational resource, because microorganisms are undoubtedly hard to see. On the other hand, some of the other organisms which are there, the invertebrates and so on, are certainly easier, although you have to look hard for them in a sense. But I think it's the kind of thing where a compost heap can bring to life the notion of an ecosystem and recycling and so on. And, the complexity of so many different types of organisms which contribute to that whole process. As I said, it's difficult with the microorganisms because, they're rather trickier to see visually. But if you take out a handful of compost and if you've got sharp eyes, but preferably, some sort of decent magnifying capacity. I mentioned in the book, you can get these really not bad digital microscopes, which you can buy off a certain well-known internet retailer, which, cost about £20. I bought myself one a few months ago. And okay, it's not a high-grade microscope, but it's not bad. You can plug it into your computer so you can kind of share what you're seeing. And, I think for kids, they'd be quite surprised when you tip out some compost onto a tray or put it in a small dish or whatever, or put some underneath one of these microscopes. You soon begin to reveal a world of organisms, which I guess most people would be unaware of in a way. That's quite exciting. It is a kind of adventure in a sense to have a look at some of these. So yes, there are plenty of invertebrates. Quite what will be there, as is true of compost in general, will depend on where the compost heap is, how it's structured, whether it's got contact with the soil, what materials in it, what the general environmental conditions are within and around the compost heap and so on. But almost irrespective of where your compost is, once you're pulling out material from the kind of established bit of the heap, you will find a variable group of invertebrates, which, as I say, with some sort of magnification, soon become very apparent, scuttling around, running around, and so on. I think you raise the issue of what contribution they make to the decomposition process. Well, curiously, even though soil invertebrates in relation to decomposition or in natural habitats, even though that's been studied really for quite a long time, it's fraught with some practical difficulties in terms of study. And there have been differing views as to what they contribute to the decomposition process. I think it's reasonably clear that they do make a contribution. Various mites, for example, will chew their way through, say, leaf material. And that chewing process in itself opens up those leaves for microbial attack. So it's a kind of communal effort in a sense, I guess. You've got the contribution of the invertebrates, which are kind of doing their own thing. They're not there specifically to help the microorganisms, but in the way that they kind of physically attack the litter by chewing their way through it. And even the feces they produce and so on, all of that makes what would otherwise be potentially quite an enclosed leaf, which often has a kind of waxy layer on the outside. It makes that much more accessible. Various figures have been bandied around, but I guess you could probably say maybe they contribute a quarter, however you measure that, but let's say contribute a quarter to the decomposition process. But of course, you've got to define what you mean by that. But that's a kind of rough and ready indication that, yes, they do have a role and they will help that decomposition process and also provide a lot of interest, as I say, from an educational point of view. And that compost heap is a self-contained or semi-self-contained ecosystem, which you can delve into and, just see what's there working in that particular type of ecosystem.
Yeah, and you can imagine how the more mobile organisms turn up, so some of the larger invertebrates, that kind of thing. But these microorganisms, how do they even know where do they come from? They just seem to kind of appear and multiply, but they must come from somewhere in the first place. Do we know?
Well, it's almost an easier question to answer for microorganisms than it is for invertebrates. Microorganisms, a lot of them are pretty ubiquitous. If you think of one example I talk about in the book, just to illustrate a fungus, for example, that people are familiar with is the penicillium fungus, which you often, if you bought your satsumas from Tesco or wherever, if you leave them in the dish for long enough, you soon get this wonderful blue bloom all over them and your satsuma or your orange is no longer edible. But that blue bloom is a mass of potentially millions of spores, which will eventually leave home and be carried along on the wind settling in various locations, including things like leaf surfaces. So, if you imagine the average kind of leaf surface is being assailed by a mass of material which is kind of drifting on the wind. So when those leaves get into the compost heap, there will certainly be fungi there. Chances are there'll be bacteria there, some of which are potentially going to live off that decaying or initiate the process of decay of that plant material. So generally they are very mobile. And, of course, when you're putting stuff into a compost heap, people will often chuck in plants that are finished, that they've pulled up by the roots. There'll be soil attached to them and so on and so on. So it is surprising how quickly one can get a good assemblage of certainly microorganisms entering that heap. And of course, as far as the invertebrates are concerned, a lot of compost heaps are laid directly on soil in the corner of a garden where there may well be leaf litter around, in some forgotten part of the garden, relegated to the less obvious parts of the garden. So things will wander in. The chances are the compost heap is not going to be animal proof, so things will find their way in. And that's not usually an issue. But as I say, the actual nature of the community of invertebrates that you get in that heap will undoubtedly be different. Not only will it be different, perhaps potentially from one compost heap in one type of location from another one, but actually if you were to take material out from different levels of the compost heap, you'll get a difference potentially in the kinds of invertebrates there. Some prefer fresher material, others perhaps more mature material. But the other thing that is discussed in the book is the issue of succession of microorganisms. So whether it's invertebrates, whether it's microorganisms, you do get this process of succession. So fresh material will have a different community of invertebrates and microorganisms than one which is more mature. And that's certainly very true of the microorganisms. Perhaps some invertebrates are slightly less fussy.
Yeah. Again, I suppose that process in reverse, the invertebrates can get out of the compost heap if they don't like the conditions, whereas the microorganisms presumably are more kind of at the mercy of what happens in the compost heap.
Well, that's very true, and that's obviously a good point, because although I don't deal with it in any great detail because it's generally much less relevant to a garden bin or garden compost heap. Of course, if you design your compost heap effectively with good insulation and so on, the temperature does rise really quite seriously within the heap. And the central parts of the heap will get hotter and hotter and hotter. And some of these invertebrates, the ones that can get away, will be driven out towards the edges, or driven out altogether potentially, or will just succumb to the heat, because in a well-insulated, large, certainly in a commercial compost heap, large commercial compost heaps, the temperature gets quite ridiculous, 60, 70 degrees centigrade. And that will bump off many of the invertebrates and also the microorganisms. You get a very different kind of suite of microorganisms in that kind of situation. But as the temperature subsequently drops, as the compost begins to go through a kind of maturation process, there'll be some reinvasion and there'll be yet another change of guard as far as the microorganisms are concerned.
And in the grand scheme of things, do we understand much about the composting process in terms of what happens inside the heap, or do you think there's still a lot to discover?
I think that comes back to the point which I made, which is that each compost heap, each compost bin is going to be different to the one next to it, to the one in the next garden, and so on and so on. So there is obviously a kind of general understanding. I don't think it would be fair to say that people don't, in general terms, understand what's going on. But in terms of a specific compost heap, in a sense, almost each one is worthy of a separate study. If you go into natural or semi-natural habitat, I don't know, if you go into something like, let's say, beech woodland, you go to 2 or 3 different beech woodlands, somewhere in the Chilterns or wherever it is, the floor is covered in leaf litter, and that's being decomposed, just as in the sense it would in a compost heap. But broadly speaking, wherever you take your sample from in that beech woodland, if you did a big study, you'd broadly understand what was happening in that beech woodland, and any sample from one piece of beech woodland would be broadly similar to a sample from another beech woodland. So you could have a fair idea as to what you'd expect to find if you'd taken one sample and you looked at other samples. For the reasons I've said, with a compost bin, it is rather more difficult. And again, you can make some generalizations. There might be groups of organisms. You might expect to find mites, for example, in terms of the invertebrates. But which particular assemblage of mites you're going to get varies, as I say, between heaps potentially, and even within the heap in terms of the different levels within it. So what the book presents, I guess, is a general picture of what goes on in compost. But in terms of an individual compost heap, that would then have to be kind of tweaked in terms of what's happening in that particular situation. So it's a beginning of an understanding, but it's not a kind of absolute thing where you can say exactly this, and this is happening.
Given that we are putting in materials that are bringing their own kind of innate microorganisms with them, and then that compost heap is very site specific, it's material specific, then the product that comes out at the end, again, is very unique. And then that product will go onto the site, presumably in a garden setting where it's being produced. And so I'm kind of wondering, if everything is relatively site specific, is it better to use materials that come from your local area and then use that compost in the site where it was created? Or would it make more sense to kind of almost inoculate another site with some other microorganisms or some other type of compost so that you get a broader variety? Or is that just don't we know that yet?
I think that's almost an impossible question to answer. I think you work with what you've got, in a sense. And because it's almost impossible to standardize things, you have to really go with what's in your locality. I personally couldn't feel that there was, beyond the addition of key nutrients like nitrogen, which we've mentioned, I wouldn't have thought that there was much value in getting too sort of concerned about quite what goes in and whether you could cross-inoculate stuff. I think particularly if the heap is in direct contact with the soil, I think things will find their way in. But gardens are such a potpourri of different sorts of materials that will go into the heap. Obviously, some are fairly standard, like grass, although even that will be a variety of species. But if you think that what scientific data there is suggests that something like 2/3 of the plants in gardens are not UK species. So it is really a real mix of things from different parts of the world and so on. But having said that, I mean, as far as microorganisms are concerned, the fact that something happens to come from South Africa rather than from the UK, in the main, doesn't make a massive difference to being decomposed because, broadly speaking, all plants are made of the same basic material. There are complications in relation to that. But, if it's a plant from South Africa that's a member of the Asteraceae, the daisy family, it'll be broadly similar in terms of its structure and composition to a daisy that you pulled up from your lawn and so on. So I personally don't think there's any great mileage in trying to adapt your compost to some kind of universal recipe for making sure you've got a bit of everything in there.
Thank you very much to Julian, and thanks to you for listening. Now, you may recall I asked for your thoughts about any other things you'd like to see me doing with the podcast, such as producing more audio content, written material, videos, etc. And I'd like to thank those of you who took the time to contact me, but sadly I've had very little response to the question. Now, I know you're out there listening because you can't do anything in the digital world without somebody logging and measuring it. So could you please let me know if there's anything else you would like from Roots and All in terms of content? If you're happy with just the podcast as it is, that's a valid option, so perhaps you could take 2 seconds to write that. Something, anything, so I don't feel so alone sat here behind this microphone. Dr. Ian Bedford is here now, talking about something that infects plants that, frankly, I feel could happily be wiped out anyway. There, I said it. I don't like them. They can all rot off as far as I'm concerned. Sorry, not sorry.
Just over 300 years ago, Charles Plumier, a French monk and botanist, discovered the fuchsia in South America. And when fuchsias eventually arrived in Britain during 1788, they triggered a new competitive craze amongst the botanists and plant breeders of the time to discover more species and to hybridise them to produce fascinating new flower forms that led to the fuchsias becoming the country's favourite plant of the Victorian era. And through to this present day, fuchsias have remained a popular plant within home gardens, where their teardrop-shaped flowers are a common sight within pots, borders and hanging baskets throughout the summer. Popular too with gardeners due to their relative robustness and resilience to common plant pests and diseases. That is, until 2007, when across southern England, gardeners started to find a mysterious new problem with their fuchsias. Flower buds were failing to open, and the new shoots were becoming strangely deformed and swollen. But the mystery didn't remain a mystery for long, since a bit of careful dissection of the deformed tissue under a microscope soon revealed the culprit, a minute arachnid mite called the Fuchsia Gall mite, a foreign invasive species that was invisible to the naked eye. First discovered on fuchsias in South America just 30 years earlier, the mite had been found to feed and reproduce within the meristematic tissue of a plant, and whilst doing so, produced a chemical that affected the growth of the plant's cells and prevented them from producing normal leaves and flower buds. And through the global trade of ornamental plants, the mites soon spread to mainland Europe, and then on into the coastal regions of southern England, from where they are now continuing to spread inland, possibly by hitching rides on pollinators, but certainly through the propagation of infested plant material. Unfortunately though, controlling Fuchsia Gall mite is not easy, since their presence only becomes apparent when deformities are seen on the plant, which is after the mites have become well established in the plant's tissue. Also, there are no effective chemicals that can be used against them. So instead, the only available option is to regularly remove and destroy all the affected plant growth during the summer months. And for the hardy fuchsias, cut them back as much as possible during autumn to remove mites that could still remain active under bud scales throughout the winter, particularly on frost-protected plants.
You can download or listen to the podcast direct from the website rootsandall.co.uk. Please also check out my Patreon, where you can make a one-off donation or take out a monthly subscription to help support the podcast. Because if you enjoy the show, please help it continue. I also have a GoFundMe where you can make a one-off donation. Even a one-off donation of £1 helps, and I'll be really grateful for your support. So please go to Patreon or GoFundMe and search for Roots and All Podcast.